In this video, we're going to be going over the structures of the sarcomere and how the sarcomeres shorten during muscle contraction. As we do this, I want you to keep in mind when you look at striated muscle, we have striated muscle in both the heart and the skeletal muscle. Shown here are skeletal muscle cells that are striated, and these are the stripes. As we go through the sarcomere, I want you to pay attention to what exactly is causing these striped appearances. What structurally is causing this? And as we go through this video, you should be able to answer the question as what makes the striations. So we look at the sarcomere. This is the contractile unit of striated muscle cells. A sarcomere extends from one Z disc to the next Z disc. And each myofibril is made of thousands and thousands of repeating units known as sarcomeres. And these sarcomeres go all the way down from one end of the cell to the other end of the cell. And we're going to see that as these thick myofilaments with their cross bridges or myosin heads grab onto the thin filaments, they're going to pull the thin filament over the thick filament, and that's where you're going to get a contraction. And so we're going to see sarcomeres are going to shorten, but the thick and thin myofilaments don't shorten during a contraction. When you look at the structures that make up a sarcomere, they consist of various bands and zones. So let's go through this. We have the A band. And this is the part of the sarcomere that contains the thick filament. So when you look here, the A band goes from one end of the thick filament to the other end of the thick filament. Well, the definition is it contains a thick filament, but we're also going to see that there's an overlap of thin filaments, and that's also going to be in that A band as well if there's any of those thin filaments that are overlapping in on the thick filament. The A in the A band, this stands for anisotropic, which is a fancy way of saying it appears dark under a light microscope. Here's a microscopic image of skeletal muscle, and you can see the dark areas. That's going to be where your A band is. So the A band, as I said, contains a zone of overlap between the thick and thin filaments, and an H zone, which contains only thick filaments. So we look here, A band from one end of the thick filament to the other. In the center here is the H zone, and that's between the two thin filaments. During a contraction, when the thin filaments are sliding over the thick filaments, that H zone in here is going to get smaller as the thin filaments are drawn closer together. But the A band stays the same length because the A band is just the thick filament. It's from one side of the thick filament to the other side of the thick filament. A portion of the sarcomere that doesn't contain any thick filaments is known as the I band. An I band just contains only thin filaments and is light underneath a light microscope, which is called isotropic. So the area here where you don't have any thick filaments, that's going to be your I band. And I always think of the light area, the second letter in light is I band. The second letter in dark, which is this area here, it's going to be dark underneath the light microscope. Well, the second letter in dark is A. That's the A band. The I band is actually part of two sarcomeres and is going to shorten during muscle contraction. Here's the I band here, and you can see it getting shorter as those thick filaments pull the thin filaments over the top of them. In the middle of the H zone is a structure called the M line, and that functions to hold the thick filaments to one another. And think about this. When those thick filaments, those myosin heads of the thick filaments aren't bound to the thin filament, what holds the thick filament in position? Well, one of the things that holds it in position is the M line. And these are proteins that hold, we can see it, they hold this thick filament to this thick filament to this thick filament, and they hold them in position for when those myosin heads need to interact with the thin filament. So the H zone has in its center the M line. And we went over this in an earlier video. Just as a recap, you have your thick filament here, and that thick filament is actually surrounded by 6 thin filaments. You can see here in a scanning electron micrograph of a myofibril, you have a thick filament, and then if you go around the thick filament, you can count 1, 2, 3, 4, 5, 6 thin filaments. If you look over here, you don't see any thin filaments on these. Why? Well, because this is a section through it, a cross section through it, that's going right through this. And so you're not getting any of those thin filaments. But over here, if you cross section through here, you're going to get the thick filament with the thin filaments around it. Look at all this mitochondria in and around the myofibril, providing all that ATP for muscle contraction. And then here we have the sarcoplasmic reticulum, which is going to release that calcium for muscle contraction. There is another protein called a titin filament, and a titin filament is an elastic protein that anchors a thick filament to both a Z disc and the M line. So when you look at this, from Z disc to Z disc, this coil-like protein, which is called titin filament, actually goes through the thick filament all the way to the other side. It's one of the longest proteins that we have in the body, and it contributes to the elastic recoil of muscles in the return to resting length during relaxation. So it's like a molecular bungee cord. Let's test our knowledge. Pause the video and name the parts of the sarcomere. Here are the answers. And cross bridges can be used as well as myosin heads. Those are both correct answers. Let's test our knowledge. Go ahead and label the following structures. And here are the answers. So where do those striations come from? Those striations come from the light and dark bands. So the A band, that's this area here. That consists of your thick myofilament and some overlapping of the thin myofilaments. Then there's a space on each side. That space is the I band. And in the center of the I band is the Z disc. This line right here is a Z disc. Over here is the next Z disc. So between these is a sarcomere. When we're looking at this one muscle cell here, these are nuclei within the muscle cell. From here to here is a muscle cell. This is the length of the muscle cell. And what do we see? We see alternating sarcomeres, back to back to back to back, all the way down that muscle cell. That's the contractile units. When you contract the muscle, these sarcomeres between the Z discs are going to shorten. The thick and thin myofilaments don't get smaller. They don't shorten, but the distance between the Z discs certainly do get smaller during a contraction. There's another protein called dystrophin, and dystrophin is a cytoskeletal protein that links the thin filaments of the sarcomere to integral membrane proteins that you find in the sarcolemma or muscle cell membrane. Here is a dystrophin protein, here's the actin of the thin filament, and it attaches to the sarcolemma or muscle cell membrane. Now in connection with associated proteins, this functions to reinforce the sarcolemma and help transmit the tension generated by the sarcomeres to the tendon. It's a real important protein for doing this. People who have muscular dystrophy, well, this refers to a group of inherited muscle destroying diseases that cause progressive degeneration of skeletal muscle. And it's characterized by the replacement of degenerating skeletal muscle by fatty and fibrous connective tissue. So here you can see this is a cross section. We have our skeletal muscle cells, but there's a lot of connective tissue in between these cells. And then this right here is adipose tissue. Duchenne muscular dystrophy is the most common form, and it's due to a mutated gene on the X chromosome. So it's an X-linked inheritance. These individuals produce little to no dystrophin protein. Therefore, the sarcolemma is going to lack support. Now, the result of this is that the sarcolemma tears during muscle contraction, and myofibers slowly rupture and die. And then they're going to be replaced by that connective tissue and adipose tissue. The disorder is apparent usually between the ages of 2 and 5. By the age of 12, a person with Duchenne muscular dystrophy usually will be in a wheelchair, and then by age 20 to 30, death can result due to respiratory failure and cardiac failure. Let's test our knowledge. Components of a sarcomere, part 1. You want to select the term that makes a sentence correct. Here are the answers. Components of our sarcomere, part 2. Select the term that makes a sentence correct and pause the video. Here are the answers. Part 3. Pause the video and select the term that makes a sentence correct. Here are the answers. When we're looking at muscle contraction, we have what's called the sliding filament model of muscle contraction. And basically this describes a movement of the thick and thin myofilaments during a contraction. During a contraction, those thick and thin myofilaments don't shorten, but they increase or overlap. The thin filaments slide past the thick filaments, extending more deeply into the A band, which remains at constant length. So we see here, our A band, our thin filaments are going to slide over that thick filament. The I bands and H bands are going to decrease in length as the Z discs are drawn closer together. The sarcomere represents the area between successive Z discs. Therefore, the sarcomere also gets smaller during a contraction. When all of those sarcomeres in a muscle fiber do this, the entire fiber gets shorter, which pulls on the attached tendon and then pulls on the bone. Voila, you have movement. So let's test our knowledge on this. In the process of contraction, the distance between Z discs gets what? And the length of the A band, what happens to that? And the length of the H zone, what happens to that? Length of the I band, what happens to that? Pause the video and answer these questions. And here are the answers. Distance between the Z discs are going to get smaller. Length of the A band is going to stay the same because that's just your thick myofilaments and any overlap of the thin filaments. But the H zone gets smaller and the I band gets smaller. Make sure you understand why. If you have any questions, you let me know. And when you contract your muscle, why does it get bigger? Because when you contract it, all of those thick and thin myofilaments are overlapping and it increases the size of the muscle. Let's test our knowledge. Pause your video and select the term that makes the sentence correct. Here are the answers. And that concludes this video, and let me know if you have any questions.